Good afternoon, good morning, good evening to everyone. Thank you for joining to our second day of the Zooming into the Community 3 meeting. So for those that were present yesterday, we already had our AIRR community General Assembly that was rich in discussion about our achievements. And also importantly, just to remind or to tell those that were not present yesterday, this Zooming into the Community Meeting 3 is also a big event for us because it's the celebration of the 10-year anniversary of the AIRR community. So today it's our second day that is focusing on scientific talks. And for this particular session and events, we decided to focus on specificity, which is one of the holy grail of the AIRR studies in general by different means. So I'm really honored to have our four speakers, Professor Antonio Lanzavecchia from Italy, Professor David Klatzmann from France, and Dr. Victor Gray from Oslo, and finally, Professor Maria Rodriguez Martinez from USA. So the idea of this session was to have two independent sessions, scientific session, one more focusing on the biological, immunological point of view and perspective of what antigen specificity means and how it is approached. And the second session after the break will be focusing on what computational approaches or nowadays can provide to this field. So the second session will be shared by Justin Barton, that I'm sure is present, but I can't see the list of participants. So we are going to start with our first speaker. That I'm not sure it's necessary to introduce in details because I'm fully sure that all of you know Professor Antonio Lanzavecchia. He is an eminent immunologist from Italy that has been working for many years on antibodies and has really an expertise in the field, and with his most late studies on neutralizing antibodies. So please, Antonio, the stage is yours, and
thank you.
We will have a nice introduction of your work and then a follow-up by questions.
Thank you for the invitation. I have to share my screen, so you should okay.
Can you share? Yes. Yes.
This.
Can you share? Yes, perfect.
Okay. All right. Okay, I'll try to give an overview of some focusing on more on the biology, of course, on antibodies, but also on some recent work we have done on CD8 T-cell immunodominance. Now, to start, I will just highlight the fact that in the naive antibody repertoire, the CDR and the CDR2 have a limited diversity. But and therefore, V gene polymorphism is very important, and we know that it can impact on the capacity to respond to certain pathogens, for instance, malaria or influenza. In the secondary response, of course, somatic mutation diversify the whole VDJ, and so we generate this huge repertoire by intraclonal diversification. Now, somatic mutations have a role in affinity maturation by selection of high affinity B cells, but also in diversifying the antibody specificities. And on the other hand, if they are not well selected, they can generate autoantibodies and rogue protein, or they can also redeem autoreactive cells by abolishing self-reactivity. So somatic mutations can really play a major role in shaping the antibody response, and we are interested to study all these aspects. And I would like to start from this relatively old study where we focused on antibodies that recognize the stem of the influenza hemagglutinin, and these antibodies bind only through the VH. The light chain does not contribute to binding, and these antibodies are characterized by VH169, but only alleles that have phenylalanine at position 54. This is a contact residue. So we also show that individuals with alleles that have leucine instead of phenylalanine cannot make these antibody responses. Now, what we have done at the time was to painfully isolate antibody and reconstruct genealogy trees, which is something that now you can do very easily by single-cell sequencing. Nonetheless, we have learned something interesting, namely that the first mutation that occurs in this genealogy tree is sufficient to achieve high affinity, but the cells continue to accumulate many mutations and up to 20 amino acid substitutions. And these results not in affinity maturations, but rather makes the initial mutations redundant. So basically, this is the UCA, the unmutated ancestor, binds very poorly to H1, but not H5. The branch point with just these two mutations binds perfectly to both H1 and H5, and a single P52A to A mutation is sufficient to achieve maximum affinity. But the interesting experiment is to show that if you now take a very mutated antibody and you revert the first two mutations, it still binds very well. So the conclusion is that the first mutation, of course, is important to immediately achieve high affinity, but there are many redundant and favorable mutations that accumulate in the expanding clones. Well, this was a simple clone. This was another complex one, but the story is exactly the same. The other example that I wanted to make is this antibody FI6, which was the first antibody that we found can neutralize all influenza viruses, group 1 in red and group 2 in blue. Can you still hear me?
Yeah.
Yes. Good. Okay. Okay. So this antibody needs both VH and VL, but when we reconstructed the genealogy tree, we found out that the UCA in this case has already high affinity and neutralizes group 1 viruses. And through somatic mutations, you generate a variant, which is FI6, that actually through a deletion of 2 amino acids in the light chain, and this variant can now neutralize not only group 1, but group 2 viruses as well. So this is an example where the mutations are not required for affinity maturations, but rather to broaden the specificity. And what was very interesting in this study is that the same sequence was found in plasma cells isolated in 4 consecutive years. So it means that there are plasma cell precursors, so namely memory B cells, that every time that they are stimulated by the vaccine can generate a burst of these plasma cells. The origin of autoantibodies has been still an open question. It's possible that it's failure of tolerance induction in developing B cells, or maybe autoantibodies are generated accidentally through somatic mutations. And the analysis of the UCA can discriminate between these two hypotheses. And so when we looked at 3 types of autoantibodies, and autoantibodies to GM-CSF in pulmonary alveolar proteinosis, autoantibodies to pemphigus, to desmoglein in pemphigus, and autoantibodies to citrullinated vimentin peptide. In all cases, we found that unmutated ancestor does not bind while the mutated antibodies do, which again suggests that the self-specificity of the antibody was generated and certainly is dependent on somatic mutations. And I would like to make a recent example that we haven't published yet about light chain amyloidosis, which is a very severe disease due to overproduction of free light chain by a malignant B-cell clone. And this may lead to deposition of amyloid fibrils in different tissues. Now, the point here is that while the antibodies are selected for binding to the antigen, the light chain dimers have a different specificities and are not selected. So as they mutate, they may become what we call the rogue protein. So the experimental approach was to express amyloidogenic light chains and their UCA and analyze their biophysical properties. And what we found, as you see, is that while FAB fragments are very stable at high temperature, this is a melting curve of different FABs, the light chain dimers almost melt at low temperature and aggregate as temperature is raised. They spontaneously aggregate, as you see here, and this is a property of the mutated dimers, but the dimers which are reverted to the germline with a single exception, they don't aggregate. These are the fibrils which are generated. And finally, these dimers are sticky and bind to, for instance, to HEP2 cells, like in this case. So we suggest that really mutations that occur in the light chains and are not kept under check by selection can generate rogue protein that can generate this disease, this terrible disease. Now, in the last couple of years, we know that antibodies can be improved or made rogue by somatic mutations, but there are now new ways to improve antibodies using language models, large language models. And this is very interesting approach that has been demonstrated in different laboratories, especially at that level of different companies. But this speaks to the fact, to an interesting idea that antibodies, in fact, are not good proteins, are not normal proteins, because they are assembled by VDJ recombination and by pairing VH and VL, and in addition, they are mutated to somatic mutations. So in this way, one can suggest that using this language model to make them more general protein-like is a way to improve their properties. And in fact, my colleagues that work to improve antibodies therapeutically, they tell me that using this language model, they can increase them, for instance, the melting temperature, the stability, and the developability of antibodies very efficiently. Now, I cannot resist by going through very quickly through some experiments some findings that we made in the last few years on the antibody response, not so much to the sporozoites. This is very interesting. These are antibodies have the potential to sterilize, to provide sterilizing immunity, but rather on blood stage parasites. And I want to remind you that P. falciparum uses about 200 polymorphic and clonally expressed genes in the blood stage. So the question was whether we could find antibodies that broadly react with blood stage parasites. And yes, after screening, identify some positive donors, we did isolate, as you see here, many antibodies that broadly stain different parasite isolates. These are parasite isolates isolated in the field, you see, and you see typically antibodies would recognize only one of these isolates, but these antibodies are very broad. Now, the surprising finding was that all the broadly reactive antibodies carry a mutated layer 1 exon inserted between the V and the DJ. And this layer 1 exon is encoded on chromosome 19. It's a collagen binding inhibitory receptor that you see here in this nice structure is appended on the tip of the CDR3. Now, basically, we went on and identified that LAIR1 antibody bind to the refins. So these are this type of 200 variant surface antigen that are expressed by malaria parasites. We found that the mutated LAIR1 domain alone is sufficient for binding to refin. In each individual, the antibodies were derived from a single expanded clone that accumulated a large number of somatic mutations. And somatic mutations do two things. One, they abolish collagen binding. If you like, this is an example of clonal redemption because this domain is self-reactive. And if you insert it in an antibody, it makes the antibody, it makes it an autoantibody. But all the clones, the cells we identified carry this mutation that abolish binding. And then there are additional mutations that modulate binding to the refins. And so this is the summary from three, in fact, 4 studies that we published. So basically, we find that in approximately 10% of malaria-exposed individuals, you find a single clone where there are insertions of either LAIR1 or LILRB1. These are 2 inhibitor receptors, which are inserted either between V and DJ or in the switch region. If they are inserted in the switch region, they are inserted with flanking splice sites so they can be spliced in. And so we have this very interesting structure here where we have an antibody with an insertion in the elbow, and you see that the structure opens up, VH and VL stay together, and you insert the LILRB1 domains here to which refin, the microbial refin binds. Of course, we also discover something quite interesting that, malaria targets inhibitory receptor. This was not known before this finding. But even more striking, we found that in European blood donors, there are plenty of DNA, templated DNA inserts that come from all the genome inserted in antibody genes. So we think that this is a rare, but it's also a general mechanism of generating new antibodies by inserting patches of DNA through what we call a patch repair mechanism dependent from non-homologous end joining. And if the inserts encode for an interesting specificity, as in the case of the malaria, then they are selected. If you want to see a complete analysis of the inserts, there is this recent paper from Catherine De La Rosa that was published in PNAS. Now, to finish on the antibodies, I want to just remind you about this study that we published some time ago where we looked using the 10x platform of memory on the structure, on the clonal structure of the human memory B cells and plasmablasts. And so what we find, to summarize, is that we were surprised to find very large multi-isotype and convergent IgM, IgA, and IgG2 clonal families. So we were expected to find IgG1 and T-dependent antigen, but this suggests the idea that there is a lot of T-independent responses or other mechanisms of clonal expansion and diversification, which are not necessarily related to the classical responses that we all study. We also found that memory B cells are very, very stable for 10, 20 years. And we found recurrent plasmablast in the steady state, suggesting that at low rate, memory B cells are continually activated. And so this supported a model of ongoing response of polyclonal activation sustaining plasma cell production. That's published stuff. Now, I want to take the last 10 minutes to switch gear and talk about T cells. And I like to say T cells, we still have old questions, but we have great new tools. And one tool is the orthotopic TCR replacement. Now, really, you can now synthesize an alpha beta receptor and knock out the endogenous alpha and beta genes and insert an alpha beta encoding construct in the T cell receptor alpha locus. This is a published methodology from a German laboratory, and you see here a very efficient insertion. In most of the cells, TCR is deleted, and here you have a new receptor inserted. So I really welcome this as a breakthrough technology, because we can now rescue exhausted T cell clones, tumor-infiltrating T cells. We can reproduce published data, which is so important for advancing the field, and we can improve eventually TCR functionality using different type of approaches, including maybe large language model. Now, the old question relating to T cells is immunodominance. And I don't have to go through this slide, just to remind you that the key here is allele-specific binding motifs, where MHC molecules, class 1 molecules, bind peptides of a given length with a specific motifs that serve as anchor in the class 1 molecules. And so what we know now is that peptide binding can be very well predicted. So you can have the sequence of your protein, and you can identify the peptides which are going to bind to those MHC molecules. But the question is whether these peptides are also generated by the antigen processing machinery, which is very complex, involve protein degradation, transport, and trimming. And so the question is, out of a complex antigen, how many peptides are going to be recognized? Now, we're taking a very radical approach, which is to clone the vaccine-responsive CD8 T cells ex vivo on day 6 after a booster immunization. It's a very small population, but we can gate on DR and CD38 positive cells. If we analyze this population, you see that they are mostly Ki67 positive, and they comprise some clonotypes which are very expanded, as you see, the two most expanded clonotypes. Now, out of 90, we can clone, of course, these cells, and out of 90 clones, 82 recognize either S1 or S2 transfected EBV B cells. Okay, and we can sequence and identify 35 different clonotypes that comprise within these 82 clones. Now then, we use, for the epitope mapping, we use data from immunopeptidomics. So we have sequenced the peptide eluted from autologous EBV B cells transfected with either S1 and S2. And we find out that out of the 82 clones, there are in fact 3 or probably 2 single specificities. So there are 2 or 3 that we call immunodominant antigenic determinants, or if you like to call them epitopes. I don't call them epitopes because, you know, the same determinant here in blue can be recognized by 30 different T cell clones. So that have different V alpha and V beta sequences. So this is a very rich database that we have and we can generate to look at not only at immunodominance, but also at degeneracy of TCR recognition of peptide MHC. So to summarize, if you predict, the predicted strong binders are about 250 in this donor, we find 29 by peptidomics, but only 3 of them dominate the response, and many clonotypes recognize the same peptide MHC complex. I will close this part by saying that, you know, we can now analyze on the CD4 and CD8 families under steady state. With these are the 2 colors, and you see that the donor, 70-year-old donor, has very highly expanded CD8 clones, as you expect. A young donor, no, not so much. But then overimposed on this, we can look at the cells that respond to COVID or to influenza vaccination, and we can really categorize and have a very interesting and the thorough analysis of the antibody response. And I want to finish with what has been a classical issue in T cell, in the analysis of T cell responses, which is the so-called memory inflation. So the continuous expansion of T cell clones that are specific for cytomegalovirus. Now, cytomegalovirus responses have focused classically on immediate early 1 antigen or PP65. But there's another antigen which is very interesting and captured our attention and has to do with HLA-E. Now, you know that HLA-E can present leader peptide of class 1 molecules, so-called VL9 peptides, to NKG2A to inhibit NK cells. Now, what the virus does is to block class 1, block transporter, and therefore class 2 HLA-E will disappear because there are no more leader peptides. But what the virus does is to upregulate HLA-E by expressing a single VL9-like peptide, which is encoded by the UL40 gene. So these are the VL9 peptides which are natural endogenous, are required for expression of HLA-E, and these are the UL40 peptide that the virus uses to upregulate HLA-E. So with this in mind, we now look at what we call the footprint of cytomegalovirus on CD8 T cells. We find that we isolate T cell clones, we stimulate the PBMC of this donor with autologous EBV cells overexpressing immediate early 1 or PP65, isolate a large number of T cell clones, as you see here, and map them, these T cell clones, on the 10x sequence of the CD8 T cells. And you see that the immediate early 1 and PP65 clones are, the response is relatively polyclonal and is made primarily by central memory cells. But the surprise was that there are 6 clonotypes that are hugely expanded and account for most, if not all, the TEMRA cells, and they recognize HLA-E with a single UL40 peptides, and these are the 6 clones. They account, one single clone accounts for 17% of total CD8 T cells in this donor. So more than 20% of the cells are have a single specificity, recognize this this UL40 peptide, and they have different V gene sequences, but they carry a relatively conserved J alpha 7 motif here. All right, so I have to acknowledge, many people that did the work in our lab that in Federica's lab in Bellinzona, at ETH, in Federica's lab at ETH, and in my new lab in Milano. And I maybe leave you with this summary slide in case there are any questions. I don't know if we have time, or I used...
We have time for questions. Any question from the audience? I have one about the last part on the TCR, maybe because this is my bias. So when you mention this immunodominance, and Jamie has a question then. So this immunodominance that you are revisiting, it's what does that imply? The virus, it's producing in a unspecific manner, finally at the end of the day, a specific response.
Yeah, that's a very important question. So there are potentially many peptides there that can bind and can stimulate immune response, but the processing machinery only generates relatively few of them, and at 29, and at the end there are 3 that dominate the response. Now we are expanding this analysis, and we show that among these 29 we find some cells that can recognize these 29, but by far, by and large, the large majority of the responding T cells is focusing on 2 of these epitopes, which is what immunodominance is about. I think this is important because for cancer immunology now, immunotherapy, you actually assume that all the peptides which have a binding motif will be presented by tumor cells, but probably will be only a fraction of those. I see there isn't. Jamie has another question.
Yes, absolutely, Jamie.
We can also take questions offline. So
that was a wonderful presentation. Can you all hear me okay?
yes.
Oh, good. And hello, Antonio. Thank you so much. That's wonderful presentation. Amazing work as always. So I wanted to ask you 2 things. One, do you know what the phenotype besides being CD8 T cells? In other words, are these in the exhausted population, and does HLA-E binding to these peptides lead to exhaustion?
No, well, we don't know. I think what we now have a second donor which is a similar phenotype, and so it's not every individual, because you can respond to these peptides only to this UL40 peptide only if you don't have the homologous gene, if they are foreign. And I should say this is a mechanism that was first discovered by the group of Lorenzo and Alessandro Moretta in Genova, and it's an old story that now we can address more thoroughly. Here you see that the cells are TEMRA. I wouldn't call them exhausted. In fact, these cells undergo inflation, so in a sense it's the opposite of exhausted. But exhausted cells, if I understand, they are present only in tissues. They don't express particularly high level of PD-1, for instance.
Well, the reason why I'm bringing it up is because, of course, with cytomegalovirus, you have in older folks, massive expansions of CD8 cells against it, and that can kind of crowd out the other populations that you need for responding to immune, other viruses and things. So that's why I was wondering about if it was helping to lead to any of that in older folks.
So the question is space. If they take space, I'm concerned. For instance, I have one big clone, which is 37% of my cells have been there for the last 25 years. And it's not specific for VL9 nor ACMV. I don't know what it is. Looks like. That's a good point. But it is, I would say, we have plenty of data supporting the general concept that with age you accumulate expanded clones. And it takes space, of course.
Yes, and so my second question was just about broadening. Going back to your antibody work, where you're showing that there's broadening occurring over time. How do you imagine that happening when you have similar affinity and a dominant clone to start out with? Are clones then going to separate, germinal centers and independently getting mutated? Like, how do you imagine it?
I imagine, what changes is the antigen, okay? So obviously this clone was stimulated by an H1 viruses and then generated a H3 cross-reactive, and then the H3 will selectively stimulate the mutated cells. And so it's, in a sense, it's all a matter of selection of the variant. But first you have to generate the variant through somatic mutation, and then this is selected by a new antigen.
So you're looking at also multiple infections.
Oh yes, well that's
okay.
what happens with
all right.
flu, of course.
Thank you.
Okay, Nicholas.
Yes, thank you very much for this wonderful talk. So I come from the field of multiple sclerosis research, and of course, when I see your data on CMV, it makes me wonder this connection between CMV, UL40, HLA-E, and EBV has, of course, come into focus over the last couple of years. Have you had the chance to look at an MS patient the same way you've looked at healthy donors?
As we would say, people in the lab are dying for this experiment. They want to do this experiment. And yes, it's very difficult. It's a lot of work. And so I guess the idea is to find cross-reactive cells, isn't it?
Yes.
Or HLA-E. So I should say we are just started to do this experiment, but I would be very happy to talk to anybody of you who has some interest in analyzing repertoires. One of the points that we were discussing in the lab is the fact that, we all consider EBV, I mean, B95H strain, but wild type EBVs are very different and very divergent. And so I think this type of question would require a very thorough approach. And I think maybe it's an idea to put together a network that would try to address this question, because you have to gather virologists, you have to, but that's a super interesting question. I have no clue.
Yeah. So I'm looking forward to this experiment that you will do now. So I'm very, very interested.
All right. Is there any other question? No? If not, thank you, Antonio. Thank you for the great presentation. And we will move to the next speakers.